Hello, everyone, and welcome to today's live broadcast, Manufacturing and Characterization of Therapeutic Exosomes, presented by Traves Antes, PhD. We are excited to bring you this educational web seminar presented by LabRoots and sponsored by Miltenyi Biotech. Miltenyi Biotech is a global provider of products and services that advance biomedical research and cellular therapy. The company's innovative tools support research at every level, from basic research to translational research to clinical application. This integrated portfolio enables scientists and clinicians to obtain, analyze, and utilize the cell. Miltenyi Biotech's technologies cover techniques of sample preparation, cell isolation, cell sorting, flow cytometry, cell culture, molecular analysis, and preclinical imaging. Their more than 25 years of expertise spans research areas including immunology, stem cell biology, neuroscience, and cancer, and clinical research areas like hematology, graft engineering, and apheresis. The company also offers comprehensive scientific support, consultation, and expert training. To learn more about Miltenyi Biotech, visit www.miltenyibiotech.com. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. Before we begin, I would like to remind everyone that this event is interactive, and we encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. And finally, if you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. Thank you. Now I'd like to introduce Dr. Antes, who is the Head of Analytical Development at Capricor Therapeutics, Incorporated. Dr. Antes' PhD work investigated the mechanisms of RNA editing directed by small RNA guides in conjunction with the RNA polymerase machinery. He expanded his training in RNA synthesis and transcription regulation with a postdoctoral fellowship at Stanford University. In his first industry appointment at Beltagen, Dr. Antes managed the gene expression group responsible for characterizing the transcriptome changes in knockout mouse models. He later worked for System Biosciences, where he was a principal investigator on independent small business innovation research grants, and later Senior Director of Product Development. In 2016, he joined the faculty at the Cedars-Sinai Heart Institute as an associate professor and became the head of analytical development for Capricor Therapeutics. His current research involves the characterization and development of therapeutic exosomes for a variety of disease applications. Dr. Antes' complete bio is found on the LabRoots website. Please join me in welcoming Dr. Antes. I will now turn the presentation over to him.
Hello, this is Travis. Welcome everybody to the webinar. Thank you for joining us. So today we're going to talk about how do we manufacture, characterize, and QC therapeutic exosomes for use in preclinical studies. So I work, I'm a faculty member at Cedars-Sinai, and I work for a innovative company called Capricor Therapeutics located here in Los Angeles. Capricor Therapeutics is a publicly traded company, so I have my standard forward-looking statements here that you can review at a later time. And so for today's presentation, don't worry if you tuned in and you hadn't heard the word exosome before. I'm going to tell you what they are and how they work. And then we'll review how to manufacture them from their host cells here. And then after we manufacture them, how do we characterize them and QC them? How do we profile their RNA cargo? And then do some surface protein phenotyping using flow cytometry. And lastly, I will describe and show you some data of some preclinical work we've done with these therapeutic exosomes. So let's start off with what are exosomes? Now, if you might notice the title of this slide says Background on EVs. So EVs are a more general term for extracellular vesicles. So vesicles can be derived from, by budding directly from the outer membrane shown here, and these are called microvesicles, or even called sometimes exosomes. Now the term exosome here means that it's derived from the interior of the cell, from the late endosome, and then maturing in the multivesicular body shown here. Now once this MVB collects hundreds of these exosomes, it then is trafficked to the surface of the cell, and these exosomes are then released in a burst from the cells. So the signaling that takes place in this whole process is just now being understood, and it's pretty exciting to figure out how these vesicles, these small vesicles, are assembled, how some are targeted for degradation, and how some of them are meant for release for signaling. So their size of these smaller vesicles range, they say, typically 30 to 100, sometimes 150 nanometers in diameter. There are some common surface markers shown here, Rab5b, or some other surface proteins, CD63, CD9, or CD81. I like showing this image after the cartoon, so you kind of get an idea of how these things are assembled. But here's some really dramatic images from this publication shown here from 2011. And here, if you look, this is a multivesicular body encircled in yellow, and then I've highlighted in this red box and kind of blew it up so in size that you can see these individual double membrane vesicles. These are called exosomes. So in step 1, the exosomes are present in multivesicular bodies, and then they assemble, like I said, sometimes even hundreds of them coming into here, and I'm showing in white arrows and also here in this red arrow. Here's a exosome being brought into the MVB for assembly and processing. The next step in their maturation is that they proceed to the surface of the cell, and then, like we talked about in the graphic, then they are released as a burst of vesicles. What are some of the features of extracellular vesicles and exosomes? Well, I mentioned that they're small, 60 to 150 diameters in size. Their proteins can vary depending upon the parent cell that built them. The common markers shown here are HSP70, CD63, CD9, CD81. Now, what also sets exosomes apart from other vesicles is that they carry a very diverse types of RNA cargo, messenger RNA, microRNA, long non-coding RNA called link RNA, or other non-coding RNA species, ncRNA. What do these vesicles do? Well, what we do know is that they modulate the immune system. So initially, what's believed to happen in tumor cells in the body is that they produce a lot of EVs that suppress the immune system, so your immune system is not going to clear out the tumor cells. So that's a bad thing. But there are other immunomodulatory features that we can use these vesicles to then hopefully treat inflammatory disorders. And we'll talk about that towards the end of this presentation. Exosomes can affect cell proliferation, and they're also involved in communication within your neuronal system. Here at Cedars-Sinai, at the Heart Institute, we're interested in treating heart failure. So heart failure can mean anything from a heart attack to an arrhythmia to other diseases of the heart. And it's the major public health challenge within the world, and it affects 5.7 million Americans every year. Half of the people who do develop heart failure will die within 5 years of their diagnosis. And it's been estimated that this disease costs the nation an estimated $30.7 billion every year for treatment and hospital costs. Now, a lot of our tissues in our body do have the ability to regenerate after injury. You know, the major example of that is your skin. If you get a scrape or a cut, you know, in a few days it seals itself over and installs new cells, and it looks either new or almost new. However, human hearts have very limited to no regenerative capacity. So if there is injury to the myocardium, there really isn't any way to fix that. So what the myocardium does is it infiltrates a bunch of these just like surface cells called fibroblasts, and they're very stiff cells, so they don't actually operate like myocardium anymore. Today, there are really no treatments that can reverse heart failure, which is a progressive condition after the initial injury. So now that we know that there's a great application for these therapeutic exosomes, how do we manufacture these? So I'm going to show you kind of an outline of how we do it, and so you can understand a little bit more about our process. So the first step is Capricor receives a call from a donor facility if someone has passed on and they've offered to donate their heart for research and therapeutic development. Then we get that call and we go and we obtain the organ from the donor, and then we create these explants, which are kind of pieces of tissue from this heart from different areas of the donated heart. We then culture them in these flasks and let them grow out a little bit further, and then we create what's called a master cell bank. And this is where, after we grow them to substantial numbers, we then can aliquot or put them in different vials and store them at -80 or in liquid nitrogen for later use. Once we're ready to then manufacture some exosomes, we take the master cell bank and then we put them in certain special media to create what we call cardiospheres. So this is a collection or a globe of cells that then are stem-like in nature. We expand them again in culture, and then we, after we replate them here, they form a layer on these flasks, and this now is your allogenic CDC or cell product that can be used either in the clinic or, in this talk, further processed to grow confluent CDCs, which means just they fill up the whole flask, and then we harvest the culture medium. Here it's indicated by CM. So in this culture medium will be all of the exosomes that these cells have secreted. We then isolate them, and then we characterize them, and then we freeze them in individual vials for use in preclinical studies. So here is a more detailed look at the production of the EVs. I just showed you a kind of an overall scheme. So after a passage 5 confluent CDC culture is obtained in these flasks, We then remove the medium, wash it a couple of times, and now, this is an important step, is we then culture the cells for 48 hours or 15 days in serum-free medium. Now, the reason why that's important is because when you typically culture cells, you put in fetal bovine serum, which is it's called FBS, and this helps support the cell's growth. Now, the problem with that is that there are just tons and tons of cow exosomes present in FBS. So we need to culture our cells in serum-free medium so that we're not adding in cow exosomes, but we're just going to collect the human exosomes made by these CDC cells. So the next step is that we pass the culture medium through a 0.45 micron filter. This is for sterilization. And then we store it at -80. And then when we're ready to make the EVs, we've been using a 100 kilodalton molecular weight cutoff, NWCO, ultrafiltration by centrifugation. And this unit has a essentially has a column and a membrane, and when you spin it, everything that is smaller than 100 kD ends up here in the bottom. Now, an exosome, although it's maybe 100 nanometers in size, it's more in like the gigadalton size. So it will not pass through, so it will stay up here in the top, and then you can just remove all the things that did filter through. And when you're ready, you just flip the column over, spin it again, and then here you end up with your concentrated culture medium containing the EVs. All right, so we've got our CDC cells. We've grown them up. We've conditioned them in serum-free medium. We've collected the media, and we concentrated it for exosome isolation. Now, how do we test what our recovery was? In this next portion of my talk, I'm going to talk about EV, or extracellular vesicle characterization. The topics I'm going to cover are some of the standards in the exosome industry of particle concentration and sizing them by using either NanoSight profiling, and I'm going to introduce a new platform that I've been using recently by a little startup company in Boston called nanoView, and they use a method where they use interferometrics, which essentially is looking at how light passes through vesicles on a slide. We also use standard electron EM microscopy. We'll look at the protein composition by Western blots and also flow cytometry. There's other ways to characterize the exosomes using biochemical assays. So exosomes carry this enzyme, acetylcholinesterase, which is an easy enzyme to measure using a colorimetric assay. So the first method that I mentioned is called NanoSight, and it's a dynamic light scattering technology. Here's a picture of what the instrument looks like. It has a computer, and then here's the NanoSight itself. Your sample gets put in through this syringe pump, and it ends up going into the instrument. So what the instrument uses is a microscope here, and then this is the sample chamber that you just filled with your exosome sample. So it's in liquid, and they're free to move about the chamber. And then the key next part is that there's a laser that comes through your sample chamber here, and every time a particle passes through the laser, it produces a signal. So exosomes are just too small to see by standard microscopy. So this uses a technology called dynamic light scattering. Every time an exosome passes through the laser, you know what? It forms this little speck here. Now, based upon the number of specks that the software sees, and also how fast it moves in the chamber, it then can tell you the size and the concentration of the exosomes in your sample. So here's an example up here in A of 3 different donors where we took explants, cultured the CDCs, and then harvested their exosomes. And what the NanoSight report gives you is a size distribution shown here by this curve, and this number here tells you the majority of your vesicles are what size. And I think it's a little fuzzy here, but you can see it's around 73 nanometers in diameter. For this donor, 81 for this donor, and I think 88 here. So there is some variability between exosome preps, but typically what we look for is something that's less than 120 or 150 nanometers, and that's your typical exosome or EV size distribution. Now, it also reports the number of particles per mil that it was able to count, and so these are 2 important pieces of information for going forward, so that you know first that the size distribution is correct, and also now you know how many exosomes per mil are actually in your sample. We sometimes go further and characterize them by electron microscopy. This first set of panels here, this is negative stain, EM, here. And so you can see the vesicles shown here in kind of this glowing sphere, and you can size them. And if you look at essentially what their size looks like when they're dried down, they're about, I don't know, maybe about 50 or 60 nanometers. And same thing by cryo-EM. Cryo means that your sample was frozen, and then it keeps the membrane intact, and you can visualize them here. But in both of these analysis, you see that in the dried form of these exosomes, they're actually a little smaller than 100 nanometers. To further characterize these vesicles, these exosomes you've isolated, there are some classic markers that you can use. So you make a protein lysate from your vesicles, and you run them on a Western blot. And here I'm showing you samples from the producer cells, the CDC cells, or their exosomes, which here I'm calling CAP 2003. So CD81 is expressed in both the cells and in the exosomes we isolated. There's another protein called periostin that seems to be highly enriched in exosomes versus their cells. And then finally, to show that you didn't have any cellular contamination, this is an antibody for a protein called calnexin, which is present in the ER of your cell and should not be present in your exosomes. So you see here, it is present in the producer cells, but it is not present in your isolated exosomes. So this tells you that you've got a really clean prep and enriched for the exosomes from your donor cells. And then the last thing is that you can characterize them for acetylcholinesterase activity, shown here. And there are some standard assay kits for doing this. It's a colorimetric assay, and you can read this on your standard plate reader in the lab. And you see that there is some variability from donor to donor based on the acetylcholinesterase activity, but they don't vary more than about 2-fold between each other. So this is a more comprehensive panel of things that we do to characterize our exosomes. Now, we're not going to do this on every exosome prep, clearly, but when we establish our method of how we isolate these vesicles, and we decide upon a defined protocol, then we do all of these assays to ensure that we're getting what we think we're getting from the culture medium of the CDC producer cells. Okay, so I've shown you NanoSight, and we did some Western blots and some acetylcholinesterase assays and some EM. Now there's this other new platform that I've been working with the founders with called NanoView, and they're in Boston. So this is a chip sensor where you have antibodies that are directly printed onto this chip, and then you put your exosome sample onto this chip, shown here, and then the antibodies will pick it up or have it stick to the chip, and then you scan the chip. So you can have a number of different antibodies on this chip. You put your exosome sample over, and then by this visualization software method, you can actually count and see the individual exosomes on this chip using a microscope. They've developed some automated software that allows you to figure out size distribution, similar to what NanoSight does, and also how many particles for a given antibody spot that it could detect. So here's some data that we got from our CDC exosomes. When we bound them on a CD63 sensor chip, shown here, you see that we get, across these 6 different sources, that we get a distribution very similar to what we see in NanoSight. However, their peak size is more like in the 50 to 60 range. Now that is more consistent with the EM studies, where they're dried down, and you see them exactly as they are from their membrane kind of encircling them. So that is CD63. We did the same thing for CD81 capture antibody, and again we see that the peak size of the particles is still somewhere around 60 to 65. And here I estimate 50 to 75. So this size range seems much more similar to what we saw using scanning EM. Now another aspect to this platform is once you do have the exosomes captured on this chip sensor, you can dry it down and then shadow them to look at them with scanning EM. So that little snapshot is shown here, and so these are individual exosomes bound by an antibody on this sensor chip, and you can figure out their size here, which seems to match up quite nicely to what we saw in these scanning profiles. So, which brings us to the question of, well, how come it's so different in this size diameter when you look at these exosomes either by NanoSight or by NanoView? Well, here I've done a drawing to try to explain how the 2 different technologies see an individual exosome. So in our NanoSight studies, if you noticed, sometimes they had a diameter of around 100 to 120 nanometers. Now, when this technology, dynamic light scattering, looks at a particle, it's seeing everything. So even what's inside here, and also its protein shell, if you will. So a lot of proteins that are bound to an individual exosome. So it actually is looking at the diameter coming from here to here. So that will be much larger than the individual particle itself between here shown in this brown gold sphere. So when you look at exosomes by scanning EM or with NanoView, you are just seeing the diameter of these vesicles from here to here. And if you notice in the data I showed you, both with EM, so cryo-EM was 90, the NanoView chip said it was around 70, and then the scanning EM was around 65. So this is probably closer to what you'll see within the inside interior of the exosome, and not necessarily looking at the protein shell. Okay, so we've physically characterized our exosomes now, and what makes exosomes pretty exciting, what actually got me interested in them originally, is that these vesicles are shuttles of RNA. So RNA is packaged into these vesicles. The vesicles are secreted, and then when they bind to another target cell, they can deliver this RNA, and this RNA can have major influences on the phenotype of its recipient cell. So how do you know what RNAs are important in your exosomes of interest? Nowadays, the first step is that you do RNA-seq. And we've chosen to use the Illumina platform because it's highly sensitive, and we don't need a whole lot of RNA in order to get a lot of information. Shown here are some donut rings of types of RNAs present in either CDC exosomes, mesenchymal stem cell exosomes, or called MSCs, that are either cultured by 15 days or MSCs cultured after 48 hours of hypoxia. And when we look at all the RNA sequences, we calibrate it to an endogenous reference, and then for these exosomes we use MIR22. Now for the exosomes you're studying, you might need to find a suitable reference marker that is more consistent for your particular exosomes. Now the ones that we study, MIR22 serves as a pretty good reference marker. And when we filter these through a heat map generator, what you see is that all of the CDC-derived exosomes cluster together, shown here in this red box, and that the MSCs stratify and form their own family, shown here. So these are very different. The CDC exosomes are very different from MSC exosomes. So we look at microRNAs, messenger RNAs, link RNAs, and also other non-coding RNAs. What we discovered just recently and published this year is a very interesting RNA that's present in CDC exosomes called YRNA. And when we treat our preclinical studies of heart attack in mice, what we've found out is that the exosomes that have more of the YRNA tend to improve what's called the ejection fraction of injured heart tissue. So the difference between non-potent and potent exosomes really depends on the amount of YRNA that it has. So if you've got more counts of YRNA, it tends to be more potent and helps repair the hearts after a myocardial infarction. When we look at here the number of RNA-seq reads from 50,000 up to 450,000 reads, and just focus on this YRNA fragment, what we observed is that in fibroblast exosomes, we call them NHDFs, which are normal human dermal fibroblasts, and then 3 different lines of CDCs derived from donors that tended to be non-potent in our ejection fraction assay, so they're not able to help heal the hearts. But now these other donors, shown here, here, and here, that had the higher amount of YRNA, tended to be much more potent in healing hearts after myocardial infarction. So what we conclude from this is that this YRNA fragment seems to be important in signaling damaged tissue. Okay, so that's RNA. Now, what about the surfaces of these exosomes? What's important about them on their surfaces for them to be taken up by target cells, and also these surface proteins themselves can be signaling molecules. So to do this in kind of a high throughput fashion, and you're just interested on the surface signaling proteins, we turn to using a really neat kit produced by Miltenyi called the MACSplex exosome kit. And this allows you to profile numerous surface proteins simultaneously in one assay. And the clever technology here is that each bead that has an individual antibody on it is also indexed. So each individual bead will have its own combination. Here as just showing an example of a ABC fluorescence. Now, when this is put into the MACSplex flow cytometer, each bead will flow to a certain region within the flow cell, and that's based on this index. And each one of these little areas that it flows to is what it means is that there's a particular antibody for each bead. So this bead has a certain marker, this one has a certain marker. And then the flow cytometer looks at the fluorescence in each one of these areas, and then that gives you your quantification of how many exosomes in your sample have that particular surface protein. So here's shown down here in say bead A, that is for I don't know, CD105, and when it binds exosomes, then it will count how many exosomes are CD105 positive, or CD63 positive, or say CD49 positive, something like that. So that's the introduction, the basics of how this kit works, and then you can look at the map for each spot and what it corresponds to. I've shown this here. You can find all this information on the Miltenyi website, but you can profile all of these different surface antigens simultaneously, because these beads will migrate to a different area within the flow cell. So I've highlighted a few here that I wanted to keep an eye on. So CD105 is an endothelial marker that we know are present on our CDC exosomes. So I was looking to see if this kit would validate that and verify that. And of course, we also know CD81 is present on our exosomes. CD63 for sure is present. And then we have just a little bit amount of CD9 on our exosomes. And this we know by doing Western blots. So these are just some markers I kind of kept in the back of my mind after we did these assays. So here's the MACSplex 8-color MaxQuant instrument shown here. It's a really cool device. You load your sample right here, and then it does the flow analysis. And over here on the right is an actual screenshot of what you see for each individual bead that migrates to its own area corresponding to a specific surface marker. And this is what it looks like. And you can see this one is pretty bright here, whereas maybe this one here isn't so abundant. And then the software quantitates all that and gives you a full report. So this is kind of hard to see, but I wanted to kind of give you a snapshot of a number of different CDC donors here, some MSC exosomes here, and the coloring corresponds to, if it's red, it was highly detected, and if it's dark green, it was a lot lower. And wanted to highlight, here's CD9 was detected, and then CD63 was detected And I actually use that as my calibration marker. So everything is calibrated to the amount of CD63 exosomes detected. And you'll see that this is, all these values will be 1 for all the samples. And then once that calibration is done, I noted a few markers of interest for my CDC exosome that I was hoping to see. And that would be CD105, which is great. I also noted CD49E was interesting. And then some of these others here, CD40, CD41B, tended to be more highly expressed here and versus the MSC exosomes here. So I was pretty pleased with the performance and the quantitative capabilities of this assay. Here's a condensed version of the very colorful table I just showed you, and I generated a heat map. And of the types of exosomes that we ran through these assays, the high abundant ones are shown in red, and the low abundance ones are shown in blue. And when you do a cluster, hierarchical cluster analysis, I was happy to see that all our potent CDC exosomes clustered here. The fibroblast exosomes just by themselves here. The MSC exosomes ended up being their own group. And here is a non-potent CDC exosome shown here. You see how it clusters differently from all the other potent ones here. And because I had these samples, I also had some iPSC cells that were differentiated into cardiomyocytes, and we collected their exosomes just to see how they would stack up. And that's shown here, called ICM-induced cardiomyocytes. And you see that they're actually quite different from our CDC exosomes shown here. Okay. So we've looked at the RNA, and now we've looked at the surface phenotype. Let's begin developing these for clinical or even preclinical applications in regenerative medicine. So, as mentioned earlier, our cardiosphere-derived cells, or CDCs, generate and secrete these therapeutic exosomes here. Us and many others have published and shown that these exosomes modulate the immune system. They actually calm it down. They're protective for cells. We call it cytoprotection, or they're anti-apoptotic, meaning death, so they're anti-death. They help cells or myocardium grow new blood vessels, called angiogenic. They also reduce this fibrosis that I mentioned earlier. When the heart tries to heal itself rapidly, this is like non-functional duct tape in your heart, and these exosomes are anti-fibrotic. And then finally, they have a regenerative capability, which means that they stimulate growth of new myocardium after tissue damage. And this is best shown here. This is a report from 2014 by my colleague here, Ahmed Ibrahim. And this is a cross-section of heart after it's undergone a myocardial infarction. And here is the left ventricle, which usually incurs the most damage. And you see how it's very, very thin here after this infarction or heart attack. And the blue color stands for the fibroblasts, or the fibrotic cells. So in the control or untreated, you see there's very little repair going on at all. When we use dermal fibroblasts, NHDF exosomes, you get a little bit of repair, but again, this is just because of the blue color, it's just fibrotic cells. So it's not going to contract and function very well. However, when you treat the hearts with the CDC exosomes after infarction, you see much greater wall built here, lower fibrotic tissue, and some new myocardium being constructed here. So we like to say our CDC exosomes tend to heal hearts after a heart attack. More data from those studies. Here I'm going to show you the left ventricular ejection fraction, and this measures the blood pumping ability of a heart. And so after a heart attack, and then days after the myocardial infarction, we measure the ejection fraction using echocardiography, when we're looking directly at the heart using sound waves. And in the vehicle, which is our PBS carrier liquid, was the same. Here's the vehicle, and here's the fibroblast exosomes. You see that after the myocardial infarction, the ejection fraction did not improve through 30 days of assay. However, in the animals, the group treated with the CDC exosomes, you see you get a nice improvement even after 15 days, and it continues up through the month of the assay. So that's showing the therapeutic capability of these exosomes. And how do we know they're exosomes? Well, something maybe I forgot to mention, but there's a drug that you can treat your cells with called GW4869, and this inhibits the cells from secreting exosomes. So it's a useful tool to just confirm and show that you're actually looking at exosomes. So we either had our CDCs treated with the drug carrier, here DMSO, or we treated our CDCs with GW4869, and then we cultured them for 15 days, collected the exosomes, and then used those exosomes in this myocardial infarction assay. So in the CDC cells treated with the drug, and then we collected the exosomes, you see that there really was no improvement of the ejection fraction in our 30-day assay. But as seen before, in the CDC cells treated with a control, that we did get improvement after 15 days and even enhancement after 30 days of the function of these hearts after a heart attack was induced. So we know that our CDC exosomes can get into cardiomyocytes, heart cells. What other types of cells will take up these exosomes? So we're interested in looking at what's called the biodistribution, or will these CDC exosomes only be taken up by heart cells, or are other cells willing to take these up and maybe be beneficial in a therapeutic way? So the types of cells we tested with our CDC exosomes are our fibroblasts again here. Now we've also tested corneal cells, this is from the eye. We looked at T cells, they're Jurkat T cells, and monocyte THP1 cells here. Now, so you add first you label your exosomes with this fluorescent dye called DID. Then you add them to these different types of cells, and then do flow cytometry on the cells to see what the fluorescent uptake was. So from 1 hour after you add the exosomes to 4 hours, and then overnight. So the amount of uptake now is measured by the fluorescent signal that these cells have acquired from the exosomes. And what we were happy to see is that our labeled CDC exosomes up to 4 hours and even overnight, shown here in blue, these are the cells that have taken up the exosomes, our CDC exosomes. You see that fibroblasts, cornea cells, T cells, and monocytes all are capable of taking up our therapeutic exosomes. So what other types of clinical areas might we be able to use these for to have some sort of clinical benefit. I've already shown you the heart regeneration assays and things we've done, but we also wanted to try, now that we know that eye cells will take them up, and also immune cells. So there is a eye disease called GVHD, or called graft-versus-host disease, and this is when people receive a bone marrow transplant because they've lost capable bone marrow, and they essentially have a new immune system. And one of the first indications of this rejection or this immune dysfunction is that a patient's eyes begin to be very painful, burning, irritation, photophobia, which means you're sensitive to light, you have pain, dryness, and there's no approved therapies other than eye drops to help, and those don't really do much for you. A little bit about this disease. 8,500 bone marrow transplants are performed in the U.S. every year. About 40% of these patients end up having this ocular form of graft-versus-host disease, and every year there's about 3,400 new cases every year. So it's a small orphan disease, but like I said, there are no treatments for this. And we thought, since our CDC exosomes seem to have benefit in damaged heart, would they work for damaged eyes? So the next step is we wanted to see, well, if we wanted to treat the eyes, how do we get our exosomes to the right area? So we tried two different ways. One is by topical, so kind of like what you would do for eye drops, just drop it on your eye. Or we would inject them. So you'd have a syringe with your exosomes in a syringe, and you would inject it into this subconjunctival space right here. And so we delivered them using either topical, shown here in green, or by injection, blue. And when I say healthy or versus alkali burn, this is how we do our eye repair model, to actually introduce a burn on the cornea for a short amount, just using sodium hydroxide. Then you remove it, and then you look at the healing afterwards. And we'll get to that. So here we just wanted to see if our CDC exosomes would go to the right area. And what we found, if we only injected the right eye of these test mice, that is where most of the exosomes were found. And interestingly, we had some that did travel to the left eye, and we had, from the injection, they ended up in the liver, but not by the eye drop method. Another curious aspect to this is that our exosomes did traffic to the heart. So to me, I think that's really interesting that even either if they're injected or just applied to the eye using eye drops, they seem to have some sort of homing beacon that they do end up going back to the heart, which is really interesting. It's a process I'm exploring. Immune cells in the spleen and in the lungs will pick up the exosomes only if you inject them, but not by topical. Same thing, the gonads and kidney, and we did not see any significant trafficking or biodistribution of our exosomes to the brain. So in our alkali burn GVHD models, there are two different assays that we look for. One, the first one is called a clinical score, and this looks for, shown here, kind of summarized, is the opacification, which means is the cornea getting cloudy, and then we look for new vessels or neovascularization, the area affected, if it's starting to heal and close, and some other parameters called aqueous flare, and this is where we look on the inside of the eyes. So our treatment groups here are going to be just our vehicle, or our CDC exosomes applied by topical, this is eye drop, or injected into the subconj injections. And after going through the clinical scoring, you see that so the higher the score, the worse that they're doing. So here's day 0, which this is normal eye function and appearance. In the vehicle-treated group that had the alkali burn, they got to a pretty high clinical score, meaning bad, and just gradually seemed to improve over time after 3 weeks. But with our either topical or our injected CDC exosomes, we saw significant improvement over the course of time over our three-week evaluation. Each day we measured all these clinical endpoints. So the CDC exosomes seem to heal eyes in this alkali burn injury model. And then more specifically, we looked at the corneal injury area and measured it. So is the injury staying the same size, or is it starting to heal and get better? And so here, the higher the injury area means that it's not healing at all. And in the vehicle group shown here, really no significant shrinking of the injury area was observed, but both with topical and our injected exosomes, we were able to see repair of this eye injury in this mouse model. So let's summarize what we talked about today. I introduced you to Cardiosphere-derived cells. These are from human organ donors. We culture them, and then we make their exosomes and EVs. These EVs can modulate the immune system. They can keep cells from dying. They're cytoprotective. They help injured tissue grow new blood vessels, called angiogenic. They reduce the amount of this fibrotic scarring after tissue injury, and they promote the growth of new myocardium in our heart attack mouse models. So they tend to heal the hearts of these heart attack mouse models. Additionally, and what we just finished, is that they also seem to have clinical benefits in ocular graft-versus-host disease models and help heal the eyes. I talked about looking at the defined surface phenotype using the MACSplex assays. That was useful for us because it helps us QC, or quality control, our exosomes that we generate from our CDC cells. And we believe that our CDC exosomes really hold great promise for clinical applications of either autoimmune or tissue regeneration in the clinic. So I'd like to acknowledge my colleagues here at Capricor, Kyle Peck, Jennifer Johnson-Mosley, Anne Sophie Ball-Ravens, Ahmed Ibrahim, and the VP of Regenerative Medicine here at Capricor, Luis Rodriguez-Barlado, for all their support. And my guys over at NanoView for helping me explore using their platform for looking at exosomes, such as George Daboul and David Freeman over at NanoView, who are really terrific to work with. And then also using the surface phenotype profiling assays, I'd really like to thank Matt McBrien specifically for helping us set up the assays and run them and also do the data analysis. He taught me a lot. And Rebecca LaFond at Milpenny for also some other data analysis. And Anne Gable today for organizing this webinar so I could tell you more about how we manufacture and characterize our exosomes for clinical development. So with that, I'd like to say thank you for your attention and time today, and we will entertain some questions.
Thank you, Dr. Antes, for that informative presentation. It is time for Q&A. If you have a question you'd like to ask Dr. Antes, please do so now. Just click on the green Q&A button at the lower left of the presentation window, type your question into the box that appears on your screen, and click on the send button. We will answer as many of your questions as we have time for, so let's get started. Our first question is, after exosome isolation, how do you store the vesicles?
That's a great question. So how we do it now is after we isolate the exosomes, we do a particle count and a surface phenotype, and once they pass our certain metrics for that as a good preparation, we then aliquot them into individual vials, and we cryo-store them either at -80 or liquid nitrogen until we want to use them. Now, when we are ready to use them, we thaw them quickly, 37-degree water bath, and then apply them in our assays, either in the cell culture or our animal models. So ultimately, what we'd like to do, and what we're working on right now, is to find out some good additives that then we would include in the formulation so that these exosomes can be stable, maybe even just at the refrigerator level, for 6 months to a year. So we're making good progress towards that. But for the studies I showed you here and some other work that we're doing right now, we store them at -80 or liquid nitrogen. But we're making progress into making a kind of an off-the-shelf therapeutic that can be just stored in the refrigerator.
What other types of tissues can be or are being used to manufacture exosomes?
Yeah, sure. So I told you about our cardiomyocytes that we get from explants from donors, and because we're interested in regenerating heart tissue, so we make our CDC exosomes. However, there are a number of other sources, and I think the most popular that you'll see in the literature and also being developed by other companies are mesenchymal stem cell explants from either adipose tissue from people or derived from their bone marrow. And they culture their MSCs and then in similar fashion to what I described, is collect the culture, the conditioned medium, and then isolate their exosomes. And those exosomes are being tested and developed for immune disorders, similar to what we're working on with our CDC exosomes.
How do you confirm or validate RNA sequences identified by RNA-seq?
Right. So in your exosomes that you are developing for clinical use or even just trying to understand them more, the method of choice right now is doing RNA-seq. Now this will give you a broad view of the RNA types and relative abundances of specific types and sequences of RNA. And in order to verify them or validate them, I develop sometimes even novel quantitative PCR assays. So then you would have your CDC exosomes, you would extract their RNA and make what's called cDNA or complementary DNA, and then use primers and the polymerase chain reaction in a quantitative qPCR instrument to then get a better quantification of the abundance of that sequence and to also validate your RNA-seq findings.
The next is a comment for you to comment on. Adipose tissue, bone marrow are common sources to manufacture MSCs and their exosomes.
Yeah, that's right. So there are a number of even some companies that are using those tissue sources to generate MSC exosomes. Maybe I didn't understand the question. Could you repeat that again?
Oh, sure. Adipose tissue, bone marrow are common sources to manufacture MSCs and their exosomes.
Yes, that's correct. Yeah. Those are the common sources because you can get a lot of cells from those particular tissues.
I really like the NanoView platform. How do I contact them?
So there's a couple of different ways you can go online. I think their web address is NanoView, NanoView, nanoview.com. And they have phone numbers and email addresses there. Or if you want, maybe you can contact our LabRoots friends, and they can. I can bridge that contact if you'd like, because I know them quite well.
So we're almost out of time. This will have to be our last question. How long does it take to set up and run the Miltenyi exosome MACSplex assays? Yeah,
good question. It really depends upon how many different exosome samples you want to set up, but they do have a way that you can use a 96-well filter plate to do, if you wanted to, 96 different samples. We typically would use half the plate, so 40 or 45 different exosome samples processed at a time, and really the prep and the binding takes just a couple hours, I'd say 2 hours or so, and then you wash and then you elute the exosomes that are now bound to these beads, and the flow analysis takes about an hour. So start to finish, you can do it in half a day, which is really great because then you end up with profiles of your 40 different exosome samples all within an afternoon. So it's very streamlined.
I would like to once again thank Dr. Antes for his presentation. Do you have any final comments?
No, I think if you're interested in exosomes, there's a news website called exosome and then -RNA.com. You can track news articles that talk about basic research of exosomes as well as clinical development, and maybe you can check that out. But I think it's an exciting time to be in working on exosomes and finding out more about what they're capable of doing and promoting regenerative medicine.
Thank you once again. I would also like to thank our sponsor, Miltenyi Biotech, for making today's educational webcast possible. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through March 6, 2018. You'll receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us. We hope to see you again soon. Goodbye.